Support Microbe TV. It's time for our annual fundraising drive. We depend on your support to produce high-quality science videos and podcasts, and now is the time to help us. If our programs have appealed to your science interests, or if they have helped you in some way, please record a brief audio or video with your phone and tell us about it. Let us know how we empower your inner scientist. We'll use it for our fundraising efforts. Send it to incubator@microbe.tv. And for more information on how you can give us your support, go to microbe.tv/contribute. This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1179. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Ooh, I'm first in the queue. This is exciting. Hi, everybody. It's 66 degrees and sunny. Obviously, Tinkerbell is out. And we're looking at 39 overnight. So we're getting towards winter. Though I must say, we're late for the average first freeze in Austin is December 1st. So we're well past that, experiencing once again the effects of anthropogenic climate change. Here we are.
It's 3 degrees C here, and it's raining. It's supposed to snow later, but nothing ever sticks. Tomorrow it's supposed to be -7 with a 50% chance of snow. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 34 Fahrenheit, feels like 22 Fahrenheit, and it's snowing, and that's 1 degree Celsius.
Snowing. Wow.
Is it a lot on the ground yet or just started?
It's the kind that's not sticking to the streets and sidewalks very much because it's so warm. It snowed overnight too, but so there's not much.
From Montreal, Canada.
Sorry. It's supposed to stop in 40 minutes, so ask me again later.
All right. From Montreal, Canada, Angela Mingarelli.
Hello, how's everyone doing? It's so much colder here. So it's 19 Fahrenheit, I think. It's -7, and it feels like 11. It's freezing. I was working at a coffee shop this morning and just walking home. That was literally 3 minutes. I thought I was going to freeze. I didn't have gloves or a hat. My own fault. But it is definitely winter now in Montreal. And the snow has been like melting. It was warm. It was like 8 degrees 3 days ago. It's been weird, the temperature. And now it's freezing again. And it's supposed to be -18 on Christmas. So we'll see.
At the coffee shop you took us to, or me to, where they said no laptops.
It wasn't that one, but that one I go to every single day.
Yeah, because they don't want you to hang around.
Yeah, today I had laptops. I was reading the papers.
You didn't say you were working at the coffee shop, did you?
Working
I
on
was,
the papers,
yeah.
Right?
Working. You were working.
Not working. Sorry, not working. Well,
at.
just like on my laptop. I also did some other things for the lab. So yeah.
I was
not a
picturing
barista.
you as a barista or something like that.
No.
A grad student. Stuff being a PhD student.
Yeah.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. And I have Kathy's weather. It's 33 Fahrenheit, 1C, and a little light snow. The snow is sticking here, but it's not expected to amount to much.
By the way, Angela, we had on our last TWiP, which just released today, an email. You know, we have these clinical cases and people write in their guesses. And someone wrote, "I'm a friend of Angela's," at the end. I guess it was you.
What was their name? Do you remember?
I'll look it up when someone else is talking. I'll look it up during the ASV spiel.
Interesting.
If you like these programs, these science programs, where you get real science, not fake science, not nonsense, we need your support to continue them. We're a nonprofit. We depend on your donations. We don't do ads or anything like that. So go to microbe.tv/contribute. We have a number of ways that you can help us out. The best way is to write a check and mail it to us, because that way 100% of it comes to us. Everybody else takes a little bit out, PayPal, Patreon, whatever. But you can do whatever you want. You can even, someone came actually from Montreal last week and dropped off cash
Oh, wow.
US dollars, which you can do. You can drop by and I'll be here and I'll say hello, give you a tour. You can sit right here and get your picture taken.
Imagine that like physical money.
I
don't even know what that is anymore.
I had visions of somebody dropping by with, a bag of corn or a couple of chickens.
Can you imagine?
A couple of chickens, right? They leave the chickens here walking around. I just want to point out, we've been telling you for a few weeks now, we have a video on our channel about why RFK Jr. is not suitable to be the head of HHS. And at the end of the video, there's a link to go and write to a website where they'll make a letter you can send to your senators. The link in the video is wrong. Okay. I fixed it in the description of the video, but it should be www.safecommunitiescoalition.org/RFK. You cannot replace videos in YouTube. If you do, they delete the old one with all the comments and that video got 20,000 views, which is pretty good. So I don't want to lose that.
You can edit your description, right?
He did.
Yeah, I edited the description. Right. You can cut out bits of videos on YouTube. You can cut off the ends, but you can't add something. You can't overlay a corrected text though. So this is the best we can do. Safecommunitiescoalition.org/RFK. The coalition part was left out. And that's my fault because I typed it in on the video. And I make a lot of mistakes. Believe me. If there are mistakes on websites and stuff, it's my fault.
You're forgiven, Vincent.
Also, I have
feel
to say,
badly.
Vincent,
I don't want to make mistakes. Sorry, what's that?
No, that I have to say, I love your new lighting because I see that you changed the colors behind you since last time I saw you. There's more purple now, and the pink
I made it more purple. Yeah,
looks very good.
Yeah, I'll keep it that way for a while until I get tired of it. All right, so the Rosenfeld Laboratory in the Division of Viral Products at CBER, FDA, is looking for a postdoc staff scientist to continue the lab's research understanding the immune responses elicited during enterovirus infection. If you're an immunologist, microbiologist, or virologist wanting to learn more viral immunology, you can email Amy at amy.rosenfeld@fda.hhs.gov for more information. So, Kathy, what's up with ASV?
Well, as we know, ASV 2025 is going to be right in Angela's neck of the woods in Montreal. And
Yay.
at a nice conference center, July 14th to 17th, 2025. The website is the main thing you need to know, asv.org/ASV 2025. So registration is now open and the early bird deadline is May 12th. So you have plenty of time, but in case you want to register now, you can do it. But what is really good thing to do now is to go ahead and renew your membership if you didn't do that when you got your reminder from the ASV office. If you're not yet a member of ASV and you want to submit an abstract in order to be to submit a travel award, you'll need to be a member, not to submit the abstract, but to submit the travel award application. And if you're interested in using the on-site childcare or ASV cares for optional other childcare fixes, then you also need to be a member for that. So now is a really good time to do that before the rush of having to do your abstract and everything all at the beginning of February. So go ahead and go to the website and do that.
And you don't have to deal with the rush this year, Kathy.
Correct. I do not.
Who's all going to come? Do you guys know yet?
Virologists.
Virologists.
A whole bunch of them.
No, I mean, of you
I
guys.
haven't checked of us.
Do you imagine
I'm planning to
virologists
go.
abroad?
Yeah.
I just checked my calendar and I seem to be free for that. So I am tentatively planning to go at this point. I haven't been for a couple of years.
It won't be cold then.
I could head on up there. I haven't
Nice.
decided, but if there's
It's
a room on the
close.
TWiV, I would.
It's
My
close.
attendance comes with no obligation for being on TWiV. I just come and bum around.
By the
That's
way,
fine.
Angela, I got an email from when I visited, when you had me visit last year or whatever.
Mhm.
I spoke with some faculty member. He emailed me and he said, this is a francophone city, so I think you should do a TWiV in French.
Ha.
But
Good luck
what?
with
But
that.
if we don't speak French and like the host
He
don't
said
speak French,
he said he would get all the guests and he would arrange it and take care of it. I'm like, I wouldn't publish that. I don't know what they're saying. I can't. Right?
it's very
You know?
It's very Quebec for him to say
yes,
that.
how very Quebecois suggestion.
Exactly.
Well, first of all, it's not going to happen
Well, yeah.
at ASV because they don't have room, right? There's just room for one TWiV and that's it.
Yeah.
Yeah.
But on top of that, I wouldn't be able to publish it because I can't publish anything I can't understand, right?
You can tell me later who that was that said that.
I'll let you know. I don't remember. By the way, it's a student in Montreal named Nathan.
Oh, Nathan. Yeah, he's my friend Nathan. He's actually studying pharmacology now. He did his master's on my floor, and now he's studying pharmacology. Yeah.
He answered one of the TWiP puzzles. Is that? Yeah. Okay.
Yeah, he sent in a letter. He's
He was a
pharmacy
TWiV.
student.
Yeah, he was a TWiV fan before I ever even knew what TWiV was. And then he was already like, once I started coming on the podcast, he was like, wait, what? I love TWiV. I love TWiV. And it was like he's like our biggest fan.
Okay, some news items. First, this is a story that's old, but we have a week delay. We have a week delay between recording and release on TWiV, so some things are old. Kennedy's lawyer has asked the FDA to revoke approval of the polio vaccine. I have a CNN article here, but you can find many, many others. So this lawyer, Aaron Seary, he actually filed this petition in 2022, and it includes other vaccines as well. And this article, by the way, starts out by saying Trump has praised the polio vaccine as the greatest thing. He said, you'd have to really convince me not to use it anymore. So this is really nonsense because, if you stop using the polio vaccine, the IPV is what's used in the U.S., by the way. OPV is already delicensed. You're not allowed to use it because it causes paralysis. If you stopped using IPV, well, to learn what would happen, you just have to look in the summer of 2022, where there was a case of poliomyelitis in a unvaccinated individual in Rockland County, New York. The virus is circulating, and this oral vaccine-derived virus, and it will infect you if you're not vaccinated. So there's no logic to not wanting to use IPV. There are no significant side effects. It prevents paralysis. The only thing I can think of is maybe most people think polio is gone. They don't even realize that it's still circulating and causing a lot of paralysis in the rest of the world. So maybe they're thinking we don't need it, but we do. We have to keep using it.
This, so to clarify a couple of things, this petition is something Aaron Seary, the lawyer, filed on 2022 on behalf of an organization called the Informed Consent Action Network, which is an anti-vax organization, and they are petitioning the FDA to withdraw approval of a bunch of vaccines, including the polio vaccine. So that's what's going on here. This guy is just a known bad actor who's, doing this kind of thing on a regular basis, and this is a past thing that he already filed, and he's buddies with RFK Jr., and that's yet another reason why RFK Jr. shouldn't be allowed anywhere near the directorship of HHS.
Also, Mitch McConnell, who had polio pre-vaccine, has said it's a great vaccine. We're not going to stop using it. Yeah. So all these people coming out in favor, it's just unreal, right?
I'm always reminded of the T-shirt that says vaccines cause adults.
Yeah, I have one of those.
Yes.
I want one of those.
Very good. I like that very much. One more. That's an article in Nature. Wuhan lab samples hold no close relatives to the virus behind COVID. So Zhongli Shi, whose laboratory in Wuhan works on coronaviruses, has done the genome sequence of all the samples she had in her laboratory. She's always said we don't have anything close to SARS-CoV-2. And so she sequenced everything, and indeed there's nothing close enough to SARS-CoV-2 to have started the pandemic if it were accidentally released. So it's not yet published. This was reported at a meeting, but I can see that some people will not accept this. Right?
Absolutely.
Well, no, they'll just say
course.
that she didn't sequence the ones that are similar to
Or it
SARS-CoV-2,
didn't
or
release
she's
those
hiding
data.
them.
Yeah,
Right.
that's
Yeah.
Right.
But, the data just piles up one after another, showing that this came from Nature. Someone wrote a comment somewhere, I don't remember which website, one of ours. She said, oh, Dr. Racaniello, scientists are divided on the origin of SARS-CoV-2. It's like, what do talking? I didn't answer her, but I'm thinking, what are you talking
No.
about? I'm a scientist. I'm not divided. None of the people on TWiV are divided. None of the virologists I know are divided. Maybe there are one or two virologists who say, well, you should keep an open mind, which doesn't help things. But the scientists who are divided, like Brett Weinstein, who has nothing to do with virology, right? Oh, by the way, speaking of Brett Weinstein, so apparently Daniel told this story on Clinical Update. Apparently his son has been in London for a few months studying, and he made friends with Brett Weinstein's kid. And the kid said to him, my dad has beef with you guys about the origins of SARS-CoV-2. Isn't that funny? He sends a message through his kid.
Wow.
Oh my gosh.
So I'm trying to find it in this article because she's moved. She's no longer at the Wuhan Institute of Virology.
Yeah,
Oh.
yeah. Where'd she go?
I'm trying to find it. I thought it was in this article, but
Hmm.
I'm not finding it. But she did move to some other place.
Okay. All right. Now we have a snippet for you. And we had a letter a couple of weeks ago requesting us to answer some bullet points before we get into our discussion to kind of summarize things, right?
Yeah. So we were discussing this in the show notes, and it occurred to me that, summarizing papers is kind of what I do. So I might as well just do a little thing here. Since I'm not the one taking the lead on this paper, I read it and I can give a little 2-minute summary. So this is a paper in Cell using artificial intelligence to document the hidden RNA virosphere. We'll get into the details in a moment, but the gist of this is we have known for a while that there are a lot of viruses out there that we have not yet discovered, because every time somebody tries a new method to look for them, they find a whole bunch more. So a major problem in surveying the virosphere is the tools we rely on most, things like PCR, can only search for viral genomes that have at least some similarity to ones that we already know. So there's this huge, what some people call the dark virome, or the biological dark matter out there that we know exists, but we haven't been able to characterize it fully. This group came up with a new machine learning algorithm, so it's AI. It takes as its inputs not only RNA sequence data, but also predicted protein structures for something called an RNA-directed RNA polymerase, which all RNA viruses must have in order to replicate. They trained the algorithm on a subset of metagenomic data, which is nucleotide sequence information from places like environmental samples. You take some water from a stream and sequence everything in it. Then they turned the algorithm, once it was trained, this algorithm was then spotting RNA-directed RNA polymerases across a wide diversity of viruses. Then they turned it loose on a bigger set of metagenomes representing nucleotide sequences that various groups have published from a whole bunch of different environments. And the algorithm found a huge number of novel RNA virus genomes that are really different from anything we've seen before. A lot of the RNA-directed RNA polymerases that it found, so that's the thing we're looking for, are highly divergent at the sequence level. We never would have found these with PCR or by looking at things that we think look like RNA-directed RNA polymerases. But they're more similar at the structural level, and so that's one thing that they included in this that had not been done before, was putting in the structural prediction of the shape of these enzymes. So that was all done in the computer, and then they spot-checked those results by pulling out and sequencing some of these bizarre genomes that they'd found of the highly divergent viruses, and they are able to pull out and sequence genomes, so it's consistent with identifying real viruses from the dark virosphere that we wouldn't have found in any other way. Those come from a bunch of different environments, ranging from ordinary stuff to pretty extreme environments. And so this is a way to illuminate the dark virosphere, if you will. It also gives us this trove of new viral genomes, or likely viral genomes, that can now be characterized. The paper is open access, so listeners can download it and follow along as we dive into it, which we're going to do now.
I want to make a very short summary, and that is
Sure.
that they looked at transcriptomes
Yes.
not genomes.
Right.
And so that makes a difference. So basically, they used really cool high-tech artificial intelligence to identify a whole lot more viruses than we ever knew about before.
Hmm.
All right, so the first two authors are Jin Hu and Yang He, and we have Eddie Holmes, Xiaorong Li, and Mang Shi as the senior corresponding authors. It comes from a variety of institutions. You've got Sun Yat-sen University, Alibaba Cloud Intelligence, Wuhan CDC, the Polar Research Institute of China, Fudan University, Shanghai, Westmead Institute for Medical Research, that's in Australia, University of Sydney, Zhejiang University, Guangzhou University, Chinese Academy of Medical Sciences, and Peking Union Medical College, the City University of Hong Kong, the Laboratory of Data Discovery for Health Limited, that's in Hong Kong, and the Guangdong Provincial Center for Disease Control and Prevention in Guangzhou. So, as you've heard, we wonder what other viruses are still out there, and RNA virus genomes are discovered in different places, environment, animals, plants, fungi, aquatic environments, etc. Usually looking for RNA-dependent RNA polymerase, which is the most highly conserved protein among RNA viruses. But there are probably many more out there to be discovered. And so this paper uses artificial intelligence approaches, particularly deep learning algorithms. And, these have had a big impact on other fields for sure. And these AI tools typically use convolutional neural networks and recurrent neural networks to do their work. These are designed for processing spatial data like images and other things to look for correlations, or to handle sequential data by capturing temporal dependencies and serial memory. But they have limitations in processing biological sequences. They have problems with inputs of various lengths, which sequences are typically, and they struggle with longer sequences. So there's a modification of them called transformer architecture. And if you know AlphaFold 2, which predicts protein structures, that's what's used for prediction of protein structures, and it garnered a Nobel Prize. It's working really, really well. So they say we can leverage this transformer architecture to try and identify RNA viruses. And so they have done that in this paper. They use a transformer-based tool. It's not oh, never mind.
Not Optimus Prime.
Yeah, not Optimus Prime, right. To find new RNA-dependent RNA polymerases. So what they do is they have 10,487 metatranscriptomes. So the RNA transcriptomes from different environments.
That would be 10,000 different samples, right?
Yes.
Different samples. Yeah, like from different environments. So that comes out to 51 terabytes of sequence data.
That's crazy. When I
This
read
is
that,
this is why one of the senior authors is from Alibaba. That's probably the heavy lifting on the data processing, and Alibaba is like the Amazon of China, so they've got the resources to build that kind of thing.
So 872 million predicted proteins. So they looked for potential RNA-dependent RNA polymerases with two strategies. One is an AI algorithm called LucaProt, LucaProt. It's a deep learning transformer-based model. It's based on both sequence and structure, as you heard, and it's based on 5,979 well-characterized viral RNA-dependent RNA polymerases, as well as 229,000 protein sequences that are not RNA polymerases. So you tell it, here's what you don't want, and here's what you do want. And these include all kinds of viral proteins and cellular proteins as well. And LucaProt was really accurate. Given this database, it was only 0.014% false positive and 1.272% false negatives. And then they applied a more conventional approach that just basically looks at sequences to do it at the same time. So by using both of these, they discover over 160,000 putative viral species, right, looking for the RNA polymerases. So that's what leukoprot identified, including 60 previously unidentified and underexplored groups that haven't gotten a lot of attention. So 512,000 contigs and 157 supergroups were identified by both leukoprot and the other approach, which is called cluster search, while 444 contigs and 23 supergroups were found only by leukoprot. So there's an overlap between the two, but leukoprot expanded it as well. They went through a number of studies to look at the sensitivity and specificity. So they compare it to other database discovery tools as well. And leukoprot always worked the best, basically. I don't want to go through them with you, but leukoprot really is the best way to do this. So 180 viral supergroups identified here. They wanted to make sure they represented RNA sequences, not DNA sequences. So they have a couple of ways that they did that. At the sequence level, they said the absence of similarity to cell proteins and the presence of an RDRP. So those two things had to be there. And that was the case for the majority of the sequences. And then, as Alan said, they extracted DNA and RNA separately on 50 of the environmental samples to look for the sequences. And only RNA sequencing reads mapped to contigs associated with viral RNA-dependent RNA polymerases, whereas both RNA and DNA sequencing reads mapped to contigs linked with DNA viruses, reverse transcriptase, and cellular organisms. So, and then a third way, they use a PCR approach showing an absence of sequences encoding RNA-dependent RNA polymerase in the DNA extractions. And then finally, they use a three-dimensional alignment to compare the newly identified polymerases with known polymerases, not just RDRPs, but DNA and polymerases of various sorts and reverse transcriptase. And the RNA polymerases clearly have a distinctive signature that's not present in the other ones. So this is identifying correctly RNA polymerases. A couple of interesting things they found. They found some very long RNA genomes in soil,
Mm-hmm.
which belong to this nido-like group, which in order is a nidovirales, right?
So it's got many different
families.
It's huge. Yeah, it's got a bunch of different families, including coronaviruses, right?
Right. Yes. And one of these genomes was 47.3 kb, so one of the longest RNA viral genomes identified to date. The other thing that's important is they find other proteins in these genomes, right? Not just RNA polymerase. And most of these, they don't know what it is, right? They have no homologs in existing databases. Right?
And they don't know what it infects.
They don't know what it infects either. Host is not known.
So this is a really agnostic approach. You're just saying what has an RNA-directed RNA polymerase and pulling out these viruses that are there. And we don't know what they're doing.
They can find signatures of structural proteins like capsid proteins and glycoproteins. They can find some signatures of non-structural proteins, but nothing really in the database. But the fact is that there are additional proteins, which shows you have a viral genome there, and that's important. So what they did was increase the number of virus species by 55.9-fold over the ICTV International Congress for the Taxonomy of Viruses.
Jens is going to be busy.
And a 1.4-fold increase from all previously described RNA polymerase sequences. They get enlargement of known virus groups and identification of novel groups that we haven't seen before. And they expanded many groups that had limited numbers of genomes. Here's an interesting fact. The supergroups with the greatest number of species now are the Narna-Levy viruses and the Picorna-Calici viruses and the Tombus noda viruses. So that's the order. So the Narna-Levy are the most numerous, the greatest number of species, followed by Picorna calissi and Tombus noda. Where are these from? All over the place. We have 1,612 locations globally, and 33% of these genomes were not described previously. So
They got a bunch of maps in this paper that are really
yes,
fun.
you could
I think
take a look at it.
it's cool that most of them, I guess the highest amount were from aquatic samples, which is unsurprising considering I remember on TWiV we spoke to some, I wasn't there, but I listened to the episode, virologists that work with like seawater, and they said that, how much was it, Vincent, per milliliter of seawater? Like hundreds of millions of viruses.
Yes.
So,
So
you can see that here.
The greatest diversity is in leaf litter. So
Hmm.
next time you're raking your leaves, folks, just
Think about
a lot of viruses in there. Leaf litter, wetland, freshwater, and wastewater environments. And the Antarctic sediment, the marine sediment, and freshwater ecosystems had the most numbers of virus particles. So the species diversity and numbers are different. And interestingly, not surprisingly, the lowest average diversity were in halite and subsurface environments. And there are not a lot of cells there.
Not a lot of cells in the rocks. Yeah.
You need to have cells.
In terms of the virus abundance, I wonder how much of that has to do with how good that environment is in preserving those
Yes.
sequences. You know, so
Yeah.
Antarctic sediment, for
Yeah.
example, I
Yep.
would think would be a good place to source sequence.
Hmm.
Yeah.
Also, that's so
and-
cool that they have samples from Antarctica. I actually looked in
Yes.
the methods because I was like, when were these collected? And it was at the Ross Sea Station in January and February 2022. And there were 9 sediments that were from Antarctica, which is cool.
Yeah, COVID, man. What are you going to do? Go to Antarctica?
Exactly.
Yeah.
Collect sequence.
Go ice fishing for viruses. Yeah.
Exactly.
So the new supergroups found in this study were mostly in aquatic and sediment samples, very few invertebrate and invertebrate animal samples. It's interesting. I don't know why that would be.
Well, I wonder how much that has to do with what the focus of sampling has been
Yeah.
before. Okay?
Yeah,
Because
yeah.
historically we focused on viruses that make us sick, right? So, who cares what's floating around in the water? Now we're a little more, open-minded about the whole thing.
Yeah. So basically, they've established an AI framework that could be used to discover RNA viruses, but they say could be extended to the description of any biological dark matter once the training data set is prepared, right? So here they had RDRPs and non-RDRPs that they used to train it with, and you could look at almost anything, and as long as you have the training material.
You could look for DNA viruses. You could look for bacteria. You could look for archaea. Yeah.
So I want to try my hand at describing my impressions of the uniqueness
Mm-hmm.
of what this adds in terms of uniqueness in the approach. And I would the one word summary is structural data, right? Whereas before we've
That's
been
2
dealing
words, but yeah.
entirely with sequence data. And why that's important in my mind, check me if I'm wrong here, or if I got it wrong, is that it's becoming increasingly evident that you can have wildly different sequences that encode similar structures.
Yes.
That is both different nucleic acid sequences, different amino acid sequences that give you similar structures, similar enough so that they do the same job. So if you're focused entirely on sequence, you're really narrowing your perspective on the thing because you're only going to see things that diverge from that sequence. Whereas if you incorporate structure into it, now that's sequence independent, really. And you expand your view. All make sense?
Yes. It's convergent evolution.
And yes, convergent evolution. And technologically, this, in the past, in searching databases and stuff, you've got raw nucleic acid sequence to deal with. You can translate that into amino acid sequence and compare the amino acid sequences, but you're still dealing with sequence. And I presume in this particular case, they're taking those same data and folding them into structures.
Yep.
Okay. And then comparing the structures. Okay. It's incredible. I said,
And the
I
only
think
reason
the-
you can do that is because of tools like AlphaFold.
yeah, I mean, it starts. It's based
Right.
on AI in the at the base of it, and then expands on that to use AI to train these programs to do these searches. Unbelievable.
Robots
So,
teaching robots.
I
Yeah.
think the paper we did with Jolene not too long ago, the pox virus and the phage,
Yep.
it was all based on structure,
Yep.
right?
Yep.
Same thing, using AlphaFold. Yep. Very cool.
So
One more thing.
my son is a computer geek, works for Google, and I sent him this paper along with Alan's summary. I don't know if he'll read it, but you know, we've been saying ever since who's the guy in San Francisco that sequences blood?
Eric
Delwart.
Delwart. Yeah.
Yeah, he was the first one that I heard talk about dark matter. Okay, we've
Yep,
got all this
yep.
sequence and we don't know what it is.
Yeah.
Okay, so this is shedding a new light on dark matter.
Do you, Mr. Jones? Speaking of AI, there's this new Google thing where you can throw a PDF into it and it makes a little podcast, a 10-minute podcast summarizing the paper. Do you know about that? It has two
No.
people. It has a
Really?
man and a woman. And Nels did it for yesterday's paper, and he played back some of it. Oh, my God. I mean, like they have the guy going, right?
Really? Oh,
Yeah.
That's scary.
And they're bantering, but they don't stray from the subject of the paper. Right?
Was it good though? The summary?
The summary was good, yeah.
I'll bet you could give the robot instructions to stray from the
To
subject. and it would do
digress,
it.
yes. So tell them something
Yeah.
about stump
Well,
grinders. Yeah.
It depends what you educate it with, right?
Yes.
Right.
There are plenty of podcasts out there that they could feed into it. And say,
Well, if they feed TWiV into it, they're going to get real wild
Yeah.
stuff. out
Rabbit
the
holes.
in
other
fact,
end, right?
that's what Nels mentioned. So the thing is, the guy sounded like an AM radio guy from the 70s, talking like that. They got to tone him down. Anyway.
What's
so
the name of this, Vincent? The software?
I don't know.
Okay, if not, I could look it up, but I need to try this.
Let's look
Now
at
I'm
the TWiV
intrigued.
notes. That's
To see.
TWiV 109. Oh, yes, here it is. It's called Notebook LM.Google.
And this is the TWiV about cats. And when did you say it'll come out?
The video's already out.
Ah, okay.
So it's Notebook LM.Google,
Interesting.
your personalized AI research assistant. So, if you're a student and you want a summary of a paper, you could do this. That would be interesting. Yeah, no, that's not the paper I'm looking for. Personally, I would read the abstract, but
Yeah.
One last thing I want to mention about this paper, as Alan mentioned earlier, we don't know what the hosts of these genomes are, right? And they have a little discussion about that. They said most RNA viruses infect eukaryotes, although that's rapidly changing. We're discovering more and more bacterial RNA viruses. But anyway, there are lots of microbial eukaryotes, right? There are tons of them in natural environments, and maybe these viruses are associated with them. But maybe they also affect bacteria and archaea as well. So we don't know. And so that's something that people can look at if they're interested. So very cool. Very good stuff. You ask, what is AI going to do for virology? Well, there you go.
That's a pretty cool contribution.
Yeah, AI keeps popping up all over the place in very positive ways. I'm sure it'll be negative as well, but
it's already got negative ways. I mean, there's one aspect of this is a lot of the AI models that people are talking about, like ChatGPT and DALL-E for artwork. The tech bros trained them on stuff that they didn't own.
Yeah.
So
Right.
they just poured the whole internet into their large language model to get ChatGPT or what's the Microsoft? Microsoft used basically all the code on GitHub to feed into their code model for their Copilot AI that can pop up unbidden, like Clippy in things you're working on. And they didn't ask anybody if they could mine these data before mining them. And there are actually lawsuits about that now because a lot of the data that went into these models that's now being resold is copyrighted. or, was open source licensed in some way, but not permission given for this. So there's already a dark side.
Okay, Alan, give us a summary of our paper.
Okay, so the paper, this is a Nature Communications paper called SARS-CoV-2 Human Challenge Reveals Biomarkers that Discriminate Early and Late Phases of Respiratory Viral Infections. So, as TWiV listeners know, a viral infection causes an immune response in the host. And we can diagnose an infection by detecting either the virus or the immune response. Classically, or recently, you might think about an RT-PCR test for SARS-CoV-2 that detects the viral genome, or at least whatever pieces of the viral genome are there. An antibody test detects whether someone's immune system has responded to that virus previously. Antibody tests are quick and easy, but they only tell us whether someone was infected at some point in the past. In contrast, there's also the interferon response, which we've talked about, which increases expression of a whole bunch of interferon-stimulated genes in response to a viral infection. It starts right at the beginning of an infection and continues throughout the infection and then subsides later. So if we could measure the interferon response with a simple test, in theory, we could detect a viral infection at the very earliest stages and maybe even track the course of it as various interferon-stimulated genes rise and fall. This group has previously been using transcriptional profiling, so they're measuring all the genes expressed in blood cells, blood's an easy sample to get, to track which interferon-stimulated genes get activated most consistently during a viral infection. That work already identified that expression of a gene called IFI27, this is an interferon-stimulated gene, is a reliable indicator of SARS-CoV-2 infection. In fact, it correlates almost perfectly with PCR test results. But those findings were all based on naturally acquired infections, so they couldn't get a reliable read on exactly when IFI27 came up. I think I might have caught this 3 days ago at the wedding, kind of thing. And figure out when other interferon-stimulated genes got turned on and off. So in this paper, they addressed that with a deliberate challenge infection of SARS-CoV-2 in unvaccinated volunteers who had no evidence of prior infections. We can and probably should talk a little bit about the ethical safeguards there as we discuss this paper. Anyway, once they'd infected the volunteers, they took blood samples at time points across the time course of infection for days so they could measure exactly which interferon-stimulated genes got turned on when and to what extent. And again, they found IFI27 gets turned on very reliably once the infection gets rolling, but they also found that another gene called MX1 gets turned on very early, in many cases even before symptoms arise and before the volunteers become PCR positive. MX1 ramps up. Then they also looked at existing data from previous challenge experiments with influenza viruses, RSV, and rhinoviruses. They found MX1 and IFI27 follow the same pattern in those infections, so this seems like a general phenomenon of respiratory viral infections. That all suggests MX1 and IFI27 expression in blood cells could be a good diagnostic test for respiratory virus infection. Might identify infections in the asymptomatic stage, and then you could use that to triage and quarantine patients for further testing and treatment. This paper is also open access, so again, you can download it and follow along at home.
So a bunch of authors on this paper, and they're all at the end.
Good old Nature, yep.
First author is Joshua Rosenheim. The corresponding author is Madad Nursadeghi. These come from University College London, Imperial College London, University of Oxford, Chinese University of Hong Kong, Wellcome Sanger Institute, and the University of Cambridge. So this challenge study...
All from the British Empire.
Yes. The challenge study was done in the UK, and many papers have been published on it. We've covered a bunch of them here on TWiV and also over on Immune. And so this is another analysis of the data to try and find if they can get immune responses that correspond to being infected, being asymptomatic or not, and being infectious perhaps. And so they look at interferon-stimulated genes. So early on in infection, interferon is produced, and the interferon turns on a whole host of genes, including IFI-27 and MX1 and many others. And so what they did in this study was to take the data and look in blood and nasal swabs for they have a panel of 26 blood transcriptional signatures associated with viral infection that they established, and that's what they use. So they have these individuals, 33 SARS-CoV-2 seronegative healthy volunteers, and they have SARS-CoV-2 put into their noses with a standardized dose, and 17 of them were infected as determined by PCR, and 16 never. They call this replicative infection. Okay.
So, my question is, they say a standardized dose for infecting humans with SARS-CoV-2. First of all, why wouldn't they say the PFU used? I don't understand. And where is that?
It's in the original paper.
Okay.
Yeah, it's 10 to the 5 PFU.
Okay.
So they pipette it into your nose, by the way. You put your head back, they pipe it into your nose, and then they clamp your nose with a clothespin for 10 minutes to make sure it doesn't drip out. Because that's how it happens naturally, right?
Yes. That's exactly what people are doing at choral practice. And
By the way,
I just want to point out for people who didn't follow this study when it originally came out, this was this was a little while ago. These folks got this done. So they're unvaccinated. They're unexposed. You probably can't find unvaccinated, unexposed people anymore. And this is a relatively young group. These people have no particular risk factors, so precautions were taken. And of course, this is being done in a clinical setting where interventions will be available if anything goes pear-shaped.
But I think they're 18 to 29, and it was it's like 80% men and 20% women, and they were all white Caucasian. Also important to know that there was very homogenous group, let's say.
It was approved by an ethics panel, but, young people predominantly get long COVID.
Yeah.
Okay?
Also.
So I don't know how you can approve a study.
I think this was, what was the date on this original study? I think it was early enough in the pandemic that
we covered this in TWiV 863.
Yeah.
Titled, "Does this challenge study pass the smell test?"
Yeah.
And the date on that was February 6, 2022.
Okay. And that was the publication of it.
That was the was our analysis.
Our analysis of the publication.
I mean, you do challenge with flu and RSV and rhinovirus because we know a lot about those viruses. We know what the outcomes. We had no clue what the outcome.
There were risks involved, and these people presumably had the risks explained, and I suspect one of the reasons maybe you got 80% male is because they were the ones who were foolish enough to do this study.
Because at that time, they didn't even know. They didn't even really know the implications. We didn't even know 2 years ago or 2.5 years ago all of the adverse reactions that
that's
are
the
adverse
point,
side
right?
effects.
If you don't know,
they tell them what could happen, but they don't even know what.
They don't even know what could happen. Yes.
Anyway, so what they do is they take a pre-challenge sample of nose and throat swabs and blood. And so they can make blood RNA and nose RNA for their subsequent analysis. Then they challenge, and then they have on day 1, 2, 3, 4, 5, 7, 10, 14, and 28, they have most of those. A few of those days they skip the nose, but they have blood on every day and nose, throat swabs and so forth. And then they use that to do RNA-seq and look for these for signatures of these genes that are turned up by the host. So that's the overall study.
I mean, nice data that can come out of this too is that we were always talking about how infection, the amount of virus put in, so PFU put in, what does that mean for viral RNA copy number coming out of the nose? And now they have all of those data. So 3 days later, if you're infected with 10 to the 5 SARS-CoV-2, then in how many people, how does it replicate differently? How does RNA copy number? They didn't do plaque assays, I'm guessing. They didn't say anything about that from the swabs, apart from the viral load. I don't think they actually did infectious virus, but that would be cool.
They mentioned infectious virus at some point. I don't know how they did it.
But I don't think they didn't mention a plaque assay.
Okay.
I think it might have just been qPCR.
So basically all these biomarkers went up in replicative infection. So those people who had PCR positivity, but they So they said most biomarkers are able to discriminate participants with and without infection. And the two of them that they end up picking, MX1 and IFI27, they have nice graphs of them. You can see if the virus is replicating, you get a good increase in these, and if it's not, it's less. Most of these, what they do is they measure the area under the curve. They have a funny name for it, which is area under the receiver operating characteristic curve, OROC.
Auroch,
Okay.
which is not the ancestor of a modern cow, but it's
So then they compared this area under the curve of each with the time point. All right. And they said most of these markers achieve near perfect discrimination of people with and without infection on days 4 through 10, which is really interesting. There's an early increase in MX1 and a delayed but sustained increase in IFI27, as Alan already mentioned. And they focus on MX1 and IFI27 because they say they're all these other markers are maybe as good, but we want to focus on these two. So the thing about this study, of course, a lot of work has been done on biomarkers and infections, but those are acquired infections. You don't know what the timing is and so forth. Here, the beauty of this is that you know exactly the timing, right? You know
Yeah,
when
I
you're
think
infected.
that's the big thing. It's synchronized.
that's the real key to this.
Yeah.
Yeah. And what I found really interesting is that they found elevated biomarkers from participants who eventually were PCR positive, but completely asymptomatic, right? So obviously the virus is reproducing in those individuals, and that reproduction is sufficient to give you an increase in these
So
transcripts.
this is like precognition of your infection. You're not even PCR positive. You feel fine. You're going to go, to choir practice and then out to a bar, and your MX1 is already through the roof.
They don't even know.
They don't even know.
So you
Also
need
be
to
interest-
you need to really tune up your meditation skills
Can
well
you
enough
imagine?
so that you know when your MX1
When your
is being
MX1 is
turned.
Talk to
There's
yourselves.
also the possibility that you know that you've been exposed to somebody.
Yeah.
Yes.
Yep. Oh, yes.
It would be interesting to see all of the cases that were replicative. Oh my gosh, I can't even say that word. You know what I mean? Those that did not have symptoms or replicating virus, but they were given the same inoculation. If there was like something similar between all of those people, were they, I don't know, a comorbidity, something different about them? Like, is it just completely stochastic? Like, what's going on there? I don't know I don't think they discussed that at all. But
They've got blood samples from them, so somebody can look.
True.
Look at that. I mean,
that would be
there's
interesting.
a lot of things that could be going on, right? They could have they could have SNPs and an immune gene. They can have
Also.
interferon antibodies. I mean, that's a big one, but that's usually in older people. But yeah, that would be interesting to know. So then they took these two genes, MX1 and IFI27, and looked at the relationship between the viral load and them, right? And they have examples of these are people who developed replicative infection. They have people who have elevated MX1 and IFI27 where they're still PCR negative, right? So I don't know what you guys think, but to me, that means the inoculum is triggering interferon, and that's turning on MX1 and IFI and all the other interferon-stimulated genes. So you don't actually need virus reproduction to get stimulation of interferon, right? That's kind of interesting. Right? They say the rise in the MX1 is usually coincident with PCR, and as we said, in some individuals evident before detection. But they say MX1 peaked before the peak in viral load, so that goes up and down very quickly, whereas IFI27 increased after detection of virus and remained elevated after viral load started to fall. And we'll see kind of why this is mechanistically in a bit. They say 5 days after virus challenge, you have this transition from a predominant MX1 response to an IFI27 response. So that tells me you could look for both of these in patients and get an idea of how long they are in infection, right? Which
Yeah.
right now we have no clue. We know when you had symptom onset, right? But you could do this and say, oh yeah, you were infected 5 days ago or 4 days ago. I think
Which
that would be
also
so cool.
means, so a major reason for looking at these data for this was in the natural infections that they looked at before, you couldn't tell exactly when somebody was infected. Now, if you use these biomarkers, you could potentially collect a lot more data from natural infections and say, oh, from your MX1 levels and your IFI27 levels, we can anticipate that you were infected,
Yeah.
on the 12th.
So let me clarify something here for myself, among other things. All these people who had these measures, an interferon response, eventually turned up PCR positive, right?
Yeah, that's right. The ones who are PCR negative,
There was a high correlation between PCR positivity and that's another way. That's not as absolute because Kathy's shaking her head here.
Well, maybe I misunderstood.
Well, that's why I'm
the
trying to
question
clarify
or
this.
yeah,
There was
what's
a
in
high
figure one?
correlation between replication as defined by PCR positivity
mm-hmm.
and turning on the interferon response. But where
Right,
the correlation, where there's a lack of correlation, is kind of when all this stuff happens. Okay, you can spike interferon, but that doesn't predict when you're going to be symptomatic, or if you're going to be symptomatic, or when you're going to show up being PCR positive. Okay, the synchrony is tighter with the interferon response than with anything else like replication or symptoms or anything like that. So you can detect an asymptomatic infection that ultimately turns PCR positive.
Correct.
Fair enough?
Yeah, some of these infections were asymptomatic throughout the course, right?
And those
And that's-
were replicative but asymptomatic.
Exactly.
Whereas the non-replicative infections didn't trigger the interferon response and didn't go PCR positive, and nothing happened.
So
Which is
one of
interesting.
the reasons that I thought about this is because we were saying that in some of these cases, it's apparent that where you're getting I don't know. I think we made the statement that you're getting an interferon response without apparent virus replication, and the conclusion was you're getting an interferon response to the inoculum. Okay, nevertheless, in all of these cases, eventually you could detect virus replication, right?
Well, in
Yes.
people in which it replicated, right? But I'm looking at the people where it didn't take, it never replicated. You should still have seen an early MX1 response, right? And so I'm looking at the two curves of MX1 and IFI27 where they're just doing time.
Which figure number are you?
It's figure 2, replicative infection, yes and no, right? So yes is red and no is blue. So there's a little bit of an increase in the MX1 in the people where the virus did not replicate. So
Really, I don't think that's statistically
Well,
significant, though.
look, there are a lot of each of those dots is a patient, right?
Right, and they're
Mm.
kind of noisy in the blue, and then they just kind of stay down at the bottom. Whereas the replicative infection is just boom right on up, and they all
Yeah, but the thing is that we pick up MX1 increase even before replication. Right?
Yes.
That's the point. So it should happen in the non-replicative people too. I don't know unless something
Or
different is
maybe
happening,
unless it's
we
not
pick
the
up-
inoculum that's triggering it.
or maybe the inoculum is completely cleared rapidly in the non-replicators for
Yeah.
whatever reason, right?
Or maybe we don't have enough samples.
Yes.
A qualification here is that we're picking up MX1 before we can detect replication.
Yes.
Yeah.
Okay.
Yep. Anyway, if they look at just people who are PCR positive, MX1 is the best. They say that MX1 is a great biomarker for PCR positivity, better than IFI27. And they also found that, here's the thing that I was talking about, Angela. They say, we found MX1 provided a better biomarker of infectiousness by predicting positive viral culture in contemporary samples. So it sounds to me like they looked at.
Okay, but they don't have
Culture.
any data about that or ever mention it again.
Yeah, I know they don't mention it. There's no
It's just like-
there's no reference in the
yeah.
text either.
By the
So-
way, audience, I have a teaching moment here. This is how science is done.
Yes.
Okay, we sit around and we read the paper and we try and understand it. We don't necessarily agree, but we try and find the truth. We try and find what's true, not who's right. Okay.
And by the way, I don't think the Google thing could do this with their
No,
No.
right.
no way. Good point.
Not yet. No, I'm joking.
No, in
Maybe
fact,
not-
people have already commented on how if you get ChatGPT answering questions and it gives questionable answers that you then call it on, it immediately backs down. Like it doesn't even it has no concept of whether what it's saying makes any sense at all.
Also, if you ask it multiple times, you get a different answer.
You can get
each time.
different answers, yes, because those models are not actual intelligence. They're just remixing the internet.
Yeah, that's right.
So, it's pareidolia. We see intelligence even where it doesn't exist.
Well, it's funny. If you ask a human the same thing 3 times too, I think you get a slightly different answer.
You can
each.
also
Every time
get
you ask
slightly
someone,
different
it
answers,
gets like,
but they will defend them each time. Yeah.
I know, yeah.
So what I wanted to say about the sample size is that they used 33 volunteers, and after they did their inoculation, it turns out that 17 of them had evidence of replication
Yeah.
and 16 did not, which
Right.
is not a fact that I think we've dwelt on very much, but I think it's kind of interesting that given this dose, it just so happened that they got 50% of
Yeah.
them.
Yeah.
And the way it was delivered, it made sure that the virus got in the nose and stayed there, right?
Right.
Well, yeah, that's what I was
Yeah,
saying
so
before. It'd be interesting to look at those people. What are the half of those people? What's different about them? What do we think is different? Does their immune profile look
So in-
different?
so
Do they
in
have
this
like more
cohort-
macrophages? I don't know.
In this cohort, which, as you pointed out, Angela, is very restricted, right?
Hmm
50% of the people get infected. What if you had a broader representation of people? Would it be 50% or it would be different? That's really interesting.
And I mean, it just, to Kathy's point, it is really interesting that they happened to pick a dose, because this was not like one of 12 live challenge experiments as with SARS-CoV-2. That was not a thing. So they happened to pick a dose that gave them an ID 50,
Yeah, that's right.
right?
yeah.
And it's entirely possible that that's just stochastic. And it may be, as Angela's saying, there may be something in the half that didn't get it, or it may just be, that's the random chance when you have this particular dose in that particular scenario.
Well, if
Now-
you infect lab mice, inbred lab mice, you get the same as you increase dose, you get more and more of the
Yeah.
mice infected.
And in
it's-
mice, you can't pinch their nose. They sneeze it out sometimes, and then half of it's gone, and you're like, "Oh my gosh!" But I wonder if in those half of the people that didn't have replicative virus, if they were where the females were there, because there was only 20% of these people were females of these participants.
Yeah.
I wonder they didn't talk about sex.
Yeah. Did they
If it w-
stratify the data with respect
Yeah, that
to
would
that?
be interesting. I don't know.
Yeah. Oh, there are many questions
Hmm.
for sure, but we're not answering them. The next thing they did was compare the kinetics of MX1 and IFI27 to a panel of genes called the stat-regulated module that other people have previously studied, and they're kind of a measure of interferon responses. And basically, they got the same result. They found similar kinetics with this stat module as they have with the MX1 and IFI27. Then they decided to do single-cell RNA sequencing on a subset of these patients, right? Because these samples, the blood and the nasal swabs have cells in them. And so they can do RNA-seq and look at the expression of MX1 and IFI27 and what cells it's in, right? So the MX1 goes up in all the PBMC subsets, and then it goes up on day 3 and then goes down on day 7, whereas IFI27 is restricted to myeloid cells like monocytes and conventional dendritic cells. So these two proteins are being made by very different cell types. So they did an experiment where they do a kind of sequencing that, which we've talked about recently, I think, which measures the openness of chromatin. chromatin is DNA wrapped around proteins in the nucleus, and the tighter it's wrapped around, the harder it is for it to be transcribed into mRNA. So this sequencing method called ATAC sequencing can measure the openness of chromatin. And they find that the MX1 gene has lots of open chromatin around it near the transcription start site, whereas IFI27 doesn't. So they say maybe this is why MX1 can get turned on really quickly, right? Because you don't have to open up the chromatin as you have to do with IFI27. Therefore, you can get that really rapid response even before you get PCR positivity. So then they take some single cells from people who were admitted to hospital with COVID, and they find similar results. And so that's kind of nice that in a real world situation, you get the same results. All right. So what about other respiratory infections? They look at, there are profiles for people infected with challenge with influenza virus, respiratory syncytial virus, and rhinoviruses, right? So they take the transcriptional data from those studies, which are available, and they say in every case, MX1 goes up before that of IFI27. This is in whole blood samples. So it looks like this kind of response is not just restricted to SARS-CoV-2, although they point out in the discussion, this is all mild COVID, right? All these individuals had mild COVID, and maybe it changes with severe COVID, right? That's something you're going to have to capture in a real world study because you can't challenge people who are going to get severe COVID. I mean, we know exactly how to do that, but you can't do it. It's not ethical, right? Just take a 95-year-old person and then you could get severe COVID. Or give them antibodies to interferon before you challenge them, right?
Well, but again, you could use these biomarkers as a way of backdating a time course of somebody's infection to get better data on what's going on.
Yeah.
It would have also been nice to look at a DNA virus because they say like respiratory viruses broadly, and all of these are RNA viruses.
Good point.
So like an adenovirus, which is very prevalent in respiratory viruses, it's part of like the common cold. It'd be interesting to see if it was similar.
And so people have certainly done challenge studies with adenos, right?
I think
I assume so.
Actually, I don't know. In the UK, maybe. Nowhere else in the world, but I don't know.
I have a naive question.
Let's Google
Is
it.
interferon turned on by a bacterial infection?
Yeah. They're bacterial PAMPs that hit sensors, That's why people were taking BCG early in COVID, remember? The bacillus, the mycobacterial.
Would you expect the same ISGs? Or, I mean, if they're different PAMPs, they might be different ISGs.
Yeah, it could be different. It's a good question. Will be interesting to know because it's useful for bacterial infections as well, right?
Yeah, I mean, one of the essential questions here is, can you, these things go on and it's a good marker for infection, but can it discriminate between one kind of an infection and another?
Yeah.
And can it discriminate between infection and vaccination?
Yep.
Oh, that's interesting.
Yeah, vaccination would be a good one. Then there's no ethical issues there.
Right.
Depending on who you talk to.
Well.
Yes, right. That's right. So they'll go on and look at other databases, right? They have various studies like household contact studies. They have community acquired infection where they have transcriptional data. In every case, MX1 goes up early and IFI27 goes up later with similar kinetics. And they end up saying, the best is to combine these two biomarkers, average expression of each one. And that way you get the early time points and the late time points, and you can get a better signature of infection. The next thing they did, go ahead.
I'm sorry, I was going to say they did find that nose swabs are not nearly as good as blood samples for this.
That's right. There's the next thing. The nose does not know as much. Yeah.
Well,
Which kind of makes sense.
You're barely getting material sometimes too. Like you have to like scrub the sides to get cells off.
And you're
in the blood, it's like all cells.
And the cells you're getting are epithelial and not so much the immune cells that appear to be the ones that are giving you these signals.
Unless you go, we did a paper on immune which showed if you go really far up, you actually get germinal center cells all the way up. Really interesting. But the nose the signal is low. The response is delayed compared to the blood. So they say blood basically is going to be the best to measure these things.
So I mean, if we're talking about a rapid test that you could do on people, best case would be this would be a finger stick, not a nose swab.
Yeah, that's true.
That's right. You can look at these two markers and get an idea of where you are in infection, and perhaps many other things. I mean, this has to be further validated with additional people, but really interesting. So basically, they have two biomarkers for infection that they temporally discriminate different parts of infection, right? MX1 is early and IFI 27 is late. So the earliest part of SARS-CoV-2, you get this rapid upregulation of MX1, which could happen even before PCR detection. That correlates with viral load measurements. And then the IFI 27 happens after PCR positivity, doesn't correlate with viral load measurements, and continues beyond viral clearance. It's very interesting. And both markers, I think we mentioned this, but worth mentioning again, both marker detection are independent of symptoms. So you can have these, and so you could be infected and you just get this blood stick and look for you have upregulated MX1 and IFI 27. You're likely contagious, so you better, wear a mask.
So I'm thinking the MX1 is the IgM of the interferon response.
Yes.
Because we talk all the time about one of the issues with administering antiviral drugs is that you have to get things in the beginning of the infection. So this gives you one more thing that you might be able to measure if you could do it simply enough to tell you that, you're in the beginning of an infection, even if you're asymptomatic.
And this is since it's so early, it's before PCR, it's before symptoms necessarily go positive. This is the kind of test we would have wanted early in the pandemic when we were going around taking everybody's temperature, which was useless because the virus is spreading before they have a fever. If you'd gone around checking everybody's MX1, which would be a little more cumbersome with finger sticks or something, but maybe that's a technology that could be used.
I went through so many checks where they pointed a gun in my head to take my temperature. Oh, my gosh. So, it's interesting. The authors say in the discussion, they think that the key conceptual advance of this work is that, so we have these two blood biomarkers that differentiate early and late phases, but they do so because of epigenetic regulation, which we talked about, the open and closed chromatin. So they think that's really a conceptual advance, that you can get MX1 really rapidly because the chromatin is open and you don't have to have any of the changes associated with opening it, which you have to do with IFI 27. So that's quite interesting. It's really it's a fundamental molecular genetic advance, right?
I wonder if it's just because MX1 had in our nasal passage, not only in the nose, I guess, but in general, we're always kind of microdosing on all these viruses and antigen. And if MX1 is always at a slightly, it's I don't know if it's turned on sometimes and it's more easily because I guess it's easily turned on, I guess I should say, than IFI 27? IFI 27, which is maybe a more specific, or you need a higher viral load, or you need more replication for it to be turned on. But MX1, just like OAS1 and things like that, are kind of innate, part of the innate immune response. That's maybe always just, I don't know, in bacterial infections, fungal, who knows, if in other things it's also turned on. So it's just always kind of there at a low level. It'd be interesting to see if in the people that didn't have replicated virus, if they also had MX1 levels, and if it was similar, if the chromatin was also open to the same amount, just at baseline, even maybe it was independent of the virus.
I think MX1 is kind of the immediate early gene here.
Yeah.
It's open and the chromatin is open because it's there to be the initial response.
But how cool is it that we can take patient samples and measure the openness of the chromatin?
Yes.
It's amazing, right? You can get functional data. I love it. The last sentence of the paper, I have trouble understanding, so maybe You guys got it. Now they write translation of these viral biomarkers to near patient platforms. I'm just picturing a table near the patient, but that's not right. Near patient platforms is now required to enable further evaluation of clinical utility and impact in prospective observational and interventional studies. What is a near patient platform?
A point of care
Point of
test.
care, yeah.
So what you'd need to do is
But you have to do a clinical trial, right?
you'd have to right. So what you'd need is you'd probably collaborate with a company that's got the GMP factory that can make a device that puts out tests, test equipment already, and they would build a prototype, some kind of a tester that you can then have at a point of care in your trial environment, and try that out in a setting where you've got I don't know, patients come in with flu-like symptoms, or maybe even just in a general practice setting where people come to their internist and you do a finger stick and see what their MX1 levels are.
Okay.
And there are obviously phases you'd go through with this. You'd initially test the prototype on people who you don't think are infected with anything, just to make sure that you get readings and yada yada yada. But the upshot would be to design a little Rich drew the analogy to a tricorder for the other paper, but a little device that any doctor or nurse can whip out in any care setting and just stick your finger and then get a reading.
And it's not at all outrageous. You go to an
No.
emergency room now, and they'll take a sample and apply it to a test that does PCR.
Yeah.
And gives you a readout on 18 different virus samples and, a few bacterial samples right while you're sitting there.
Yep. And there are nucleotide probe chips and devices that will find particular, transcript is elevated. I think that's completely doable technology. And again,
here
you'd
they
collaborate with a company that does this sort of
Here
thing.
they did nucleic acid detection, right? So transcriptomics, right? So what are they going to do in the near patient platform? They're going to look for nucleic acid or protein or what? They can do PCR.
Nucleic acid, I think, is the way to go.
So they do a PCR assay for these
Yeah,
things?
use a transcript chip, but it's a transcript chip that's only got like 2 things on it.
Yeah.
Wait, don't you want to know the level of the protein? That's the bottom line, not the level of the nucleic acid that encodes the protein.
Hey, that's a good point. I don't think they ever looked at that here, did they? This is all transcriptomics.
It's all transcripts.
It's in seeks.
I
good
put
point.
into, so UM has their own proprietary GPT. I asked, what does near patient platform mean? Because I had also put it first just directly into Google, and Google said, doesn't appear like we have enough information to answer that. But the GPT is kind of what Rich and Alan said, refers to medical equipment, technologies, or systems that facilitate testing, diagnosis, monitoring, or treatment of patients close to the point of care in settings like clinics, doctors' offices, or even at home, as opposed to traditional central lab or hospital settings, enabling rapid and accurate decision-making by healthcare providers, improving patient outcomes, and increasing convenience and accessibility of medical care.
My goodness, this is a loquacious app.
Such as point-of-care testing devices, portable imaging systems, and telehealth solutions.
Oh, so near patient is a thing,
Yeah,
Yeah.
apparently.
obviously.
Well,
And
not
I
to
didn't-
Google, but to UMGPT, yes. But
Yeah,
it's a phrase that these authors use, so they didn't just dream it up.
I think point of care test is the more common
Yeah.
Okay.
phrase. And
Nice.
looking further ahead, you might even get to a point where you could incorporate this and a bunch of other transcript levels and maybe some other things into some kind of a wearable device. So your Apple Watch model 23B7, whatever they come up with next, would tell you, hey, you're coming down with a cold.
God.
But at
You
that
want
point,
to
would you
close
really
your
wanna,
rings before it hits you?
Would you want to know? Because then imagine every time, who knows how many infections that we have that don't actually reach symptoms, to have symptoms, and then you're always living on alert,
I
and you're
think we're
always
gonna
worried
find
that
out.
you're going to get sick. Like this morning, I woke up and my throat hurt. and I've been trying. Well, not in the end, it was fine. But I mean, it was fine. Obviously, there's something there. But like, I'm not going to stop living for that reason. Or yesterday, if I would have had some flashing thing, I'm like, oh, maybe I won't go to work, or I'll plan something else. And do I really want to live my life like that? Personally, probably not. It's cool. The data's cool, but like that's like people that wear the constant glucose monitors that don't have diabetes. They just want to see like their glycemia levels all the time. It's a little bit extra, but
I know many people who carry pulse oximeters along, those little things you put on your
yeah.
finger.
Also,
Okay, I think we can do a couple of emails here. Alan, can you take that first one?
Sure. Let me just get my stuff together here. Stephen writes, this week, not for the first time, I listened to your mini rants about the noise of leaf blowers. But have you considered the potential for these damnable devices to spread dangerous microbes in addition to the health-harming noise and 2.5 particulate, both from the inefficient engines and the material being blown? Consider the composition of the vast amounts of particulates and how much is from sources such as fecal matter. Studies show this stuff remains in the air up to 3 days. Speaking of transferring H1N1 from birds to pigs, you say, if it gets in their snout. Is this a way that can happen? I would like to hear your thoughts on the potential spread of disease by these machines. I live in a condo development surrounded by more of same, and the lawn maintenance contractors employ them nearly all day, every day. Steve, who's in Mechanicsburg, Pennsylvania. Steve, as you probably know, I so feel your pain. And yeah, this is yet another reason why these things are awful. I mean, my biggest complaint is the noise, because I don't have to be outside when they're blowing this stuff around. And frankly, the dilution of the particulates is probably, not going to spread a whole lot of additional disease. But yeah, sure. They're blowing poop around.
But it's animal poop. It's not human poop for the most part.
Oh, well, you can get things from animal poop.
well, true, but it's also so does the
Mostly.
wind. The wind can
Yeah,
blow the leaves,
that's
so I
what
don't
I'm
know if the
thinking.
But
risk
the wind is not
higher than the
200
wind.
miles an hour aimed at a focused point, right?
I mean, if you get leaves in your mouth more
Yeah.
frequently, maybe.
So-
But like,
The idea of the potential spread of disease, this whole thing reminds me of the hand blowers in
Yes.
public restrooms, and there have been studies about that.
That blows it, yeah.
And
those I don't actually use because of those
I hate
studies.
those.
I refuse.
well,
I'll just
but
do
if
this.
you're in there and that's all there is, so I tend to carry some kind of paper towel thing in
I
my
just
back
shake
pocket.
my hands and
I
walk
shake
out.
my hands too, and I
Yeah.
use my pants.
But if other people turn them on while you're there, there's not a lot you can do
It's
about
true.
what's. You still
So
have to breathe.
it seems to me that if there were a significant association of leaf blowers with disease transmission, we would know it, right?
How?
'Cause by association of people who get sick with leaf blowing. You'd have to do the studies, right? You
Yeah.
couldn't just say, but someone would have to say, "Oh, look, this person could come down with this infection, unusual infection." The CDC comes in and they trace it to leaf blowing, something like that. It's never
I
happened.
guess you
right?
could go in like an aged neighborhood or near a retirement home or something, and if there's people that are potentially more immunocompromised that are around leaf blowers all the time, are they sick more?
Why don't we
often?
do an experiment? Just eliminate all leaf blowers and see if infections go
Good
down.
idea.
Can you imagine?
So,
And if
Wouldn't that
infections
be
don't go
infectious
down, then just don't bring the leaf blowers back anyway.
Ever. Yeah.
TWiV 386, April 24th, 2016, titled The Dolphins Did It. The TWiV team discusses the evolution of ancient retroviruses and a discussion of virus dispersal during different methods of drying hands.
Mhm.
Yeah.
Because we've done this before. They've done experiments
Yeah.
with like M13,
Mhm.
Yeah.
the bacteriophage, to see what the dryers do. And I think there's a paper here.
They blow it
Yeah,
all over
those
the place.
those Dyson things where you put
Yeah.
your hands in it.
Blade.
They
Yeah.
blow it all over the place. Yeah, and I'm sure the
That's
leaf
gross.
blowers do the same thing.
And I want to call out New York State in particular, which has, I thought this was just in Penn Station, but I've now discovered in driving across New York State, it's also in all the rest stops on the thruway. They have put a hand drying blower as an extra faucet in these modern sinks that they
Yeah,
have.
Yeah.
right.
You must have seen these in
Oh,
Penn State.
yeah,
Yeah.
I've seen those.
So you've got
I've seen those.
it's this very elegant modern design of the sink, and there's a faucet, and right next to it there's something that looks like a faucet, and that's your soap dispenser, and there's another of the same thing, and that's a hand dryer blowing directly into the sink where somebody with dirty hands has just washed them. It's just
Mhm.
complete disaster.
All right, Angela, take the next one, please.
Yeah. Lena writes, "Hi, I'm a public health physician who has been learning a lot from the TWiV podcast. Thank you for doing it. I'm emailing because there is a listener letter that I remember from a TWiV episode that made a great impact on me and which I think about it from time to time. I can't seem to find which episode it came from, so I'm wondering if you would be able to help me track it down. The letter was about politics as it relates to science, and the gist of it was that science is by its very nature political, and that one of the great challenges of our current times is a shift from scientific experts being the one to lead the narrative around science to the masses leading the narrative. The letter writer had likened it to the Protestant Reformation. I think this letter captured in words something I've sensed but didn't really know until I heard it in the letter. I've been thinking about it ever since. I'm not sure if it rings a bell or not, or if you would be able to help me with finding this in the podcast. Thanks, Lena."
Lena, if
I
you're
have
watching
no
this,
idea.
you will see that Alan and Kathy and Vincent are very busy surfing the internet looking for your letter.
I got 4 hits for clinical updates with Dr. Daniel Griffin, but I'm sure those are not
Oh.
right.
Now, it was
Alan, you're silenced.
quite a long letter, and then subsequently someone wrote kind of a counter.
Yeah, so I found in the show notes for TWiV 1155 the letter
Yeah.
from Chris. I don't know. Did we read that on 1155?
I don't know.
Because I have other hits.
We're working on it, Lena.
71, 73, 55. Okay, so I think the
It could be 55.
I think the 1171, 73, 75, and 79 is this episode, are all this letter asking about that. So I think it's 1155 that we read it. That show was Spillover in Stall A.
All right.
But if you click on letters read on TWiV 1155, you get a hit for TWiV 1157 letters. So,
We could always reread the letter right here. Then Lena would have it.
well,
Yes.
that's if we can find it.
Oh,
I found
thought-
it in the show notes for 1155.
Oh, you found it. Okay.
Shall I go ahead and read it?
Oh, it's on 1155. No,
It's
too
on
long.
1155. Okay.
Yeah, it's too long, but it's in 1155. Yeah, if you type in the right number, and now I have to fix that. Okay.
Sorry.
No, it's okay. I make all kinds of mistakes like that. It's by Chris. Yeah. Kathy, can you take the next one?
Oh,
No, you
I
don't
would
wanna
have
do
to
it?
find today's show notes.
All right, don't worry
Okay,
about
no, here I am. Bree
Yeah.
writes, "Hi, thanks for your great podcast. I wanted to provide some more information about why microRNA is useful. The really key thing is that it offers a level of post-transcriptional regulation that can allow for the fine-tuning of protein expression as well as coordinated changes in expression. Here's more info, and I'm super happy to answer any questions you might have. I did my PhD on Argonaut, and there's 2 things that I have not checked on. One is a YouTube, and the first one is redirecting me to something about microRNA, RNAi. Oh, it looks like a blog post from 2021. So lots of information here about RNAi and microRNAs. Thank you, Bree.
Let's see. Rich, can you take the next one?
Yeah. Rodney writes, enjoy your programs as they allow me to spend Sunday mornings listening to a great discussion on a long-time topic of interest. Rodney, somewhere in Kansas. So that's just a nice compliment. He's got, that's about it, right?
There's also a story that he points out that's pretty interesting.
story that is a DNA test showed my father wasn't my father. Here's why I'm glad. Which I haven't looked at. That's interesting. And another link that's sorry. Yeah. Oops.
Oh, it's because somehow there was an umbilical cord donation that confused it, right?
Yeah. There's another YouTube link to DNA shocker, how a stem cell transplant created a false father match.
Yeah.
Okay, so those would be interesting to look at.
That's very interesting. Yep. All right, last one's from Mark. Hi, folks. I'm listening to your episode about the New Zealand experience of RSV during the COVID lockdowns and other measures. First, we are lots of islands, two main ones, North Island and South Island, a.k.a. Te Ika o Maui, the fish of Maui, and Te Wai Punamu, greenstone water. Then there is Stewart Island, a.k.a. Rakiura, glowing skies, and lots of smaller ones. I had no idea. Most people in the USA have no idea where we are, perhaps partly because New Zealand is left off many maps. Anyway, you mentioned Gemma Geoghegan, and as I heard her on the radio earlier, I thought I would flick you a link. Okay, and there's a link to an article by her. Bird flu outbreak potentially wider than yet known. Otago University virologist Gemma said bird flu spreads very easily and there's a high risk of it being passed on. So that's cool. Hey, we talked about her. The interview I heard is linked there from. You can contact Janet through this link. I think we were talking to her. On a bit of a tangent, I trust you folks have an eye on the unidentified respiratory virus killing many people in the DRC. So it turns out it's malaria. It's not a virus. Daniel talked about this yesterday with respiratory symptoms. It's spread by mosquitoes as usual, but he said due to the malnutrition and poor conditions in the country, malaria often has respiratory symptoms and it's lethal. So that's what's going on there, unfortunately.
By the way, circling back to Bree's letter,
yes.
One of those links was to a blog, and that blog is by her. She's the bumbling biochemist, okay, who I think we've identified in the past.
Yes, definitely.
And I just started playing this radio interview, but it hasn't yet introduced her so that we could figure out how to pronounce her last name.
Yeah.
Oh.
Okay, let's do some picks. Angela, what do you have for us?
So I picked from Science Magazine the breakthrough of the year 2024. And the first pick, or I guess the biggest breakthrough was this new drug for HIV, so an antiviral. It is called, let's see if I pronounce this right, lenacapavir. Is that how you guys would say it?
Yep.
So
Sure.
So it's a new drug that is injected subcutaneously every 6 months and has shown to be very effective. Especially, well, there's two different studies that have been done now. One was in cisgendered women, where I think I wrote down specifically. I think they had over 5,000 participants who were initially HIV negative, and then during this trial that they did, which I won't go into the details, but we can link the papers in the show notes. There were 55 infections, HIV infections observed out of over 5,000 people. And the people that had the lenacapavir specifically didn't acquire any HIV infections. So in that case, it showed to be 100% effective. Then there was also another trial that was with men, different one from the New England Journal of Medicine, that had 3,000 participants. And I think that there were 2 participants in the lenacapavir group that were infected. But overall, it's just showing increased protection against HIV infection when you compare it to other PrEP drugs, the prophylactic drugs that people are commonly taking, like, what is that one called? It starts with a C. Vincent, you probably know. Cabotegravir, I believe, which is a new drug that's actually taken every 2 months, because then there's also the other prophylactic drugs that people take orally every day, but that haven't been shown to be as effective. So this is just, I think, in the field of HIV, potentially a better treatment because it's literally twice a year. You don't have to worry about taking a pill orally. Also in areas where they don't have access to having oral medication, if they can get an injection, places like in Africa where HIV is still very rampant among women and men, cisgender, and all different, I guess, what's the word? I'm trying to use the specific
All
word.
different groups.
All different groups, yes. I blanked. But yes, because in the United States, like oral PrEP is a lot more available than it is in other parts of the world. So something like this, I think, is great, is a great option. And we'll see how much it costs. I think it still said that it was a little bit in the Science Magazine article said that the cost has not been released. So it's probably expensive, I'm guessing,
Sure.
but hopefully with time it'll go down. But it's a big win.
It's an interesting mechanism. It locks the capsid.
Yeah.
Yeah, which is different compared to the other drugs,
Totally
right? They
different
haven't
from
targeted
everything
the
else.
capsid.
Yeah, it's great.
And it's got an extraordinarily complex structure. There's a Wikipedia page on this. I'd be interested to know where it comes from. It says it was developed by Gilead, but I'm thinking not from scratch. I wonder if there's a natural compound behind this somewhere. Because
Maybe.
you couldn't make this one up, I wouldn't think. Maybe GPT did it.
Thanks, Angela.
No problem.
Yeah.
Kathy, what do you
cool.
have for us?
Oh, it's just a fun, quick one. It's Bach's Toccata and Fugue on a floor piano. And then I sent it to someone, and then they sent me the link to the famous Tom Hanks
Yes.
scene in Big at FAO Schwartz. And then FAO Schwartz Big Piano is back. This is a story from June 2024, but I didn't know it had been gone, but evidently it's back. So there's a lot
FAO
of hits.
Schwartz was gone.
Okay. So this is a reopening of FAO Schwartz Toy Store in Rockefeller Center. Yeah. So I don't know about that. Maybe this is a AI hallucination.
All right. Thank you. Rich, what do you have for us?
So this is an elaboration on something that came up in a recent previous TWiV. It's an oldish book and movie called Never Cry Wolf that is one of my favorites. The author is Farley Moat, and it describes, it's important that there has been some pushback into the authenticity of this. And in response to some of that pushback, Moat himself says, I took some pride in having it known that I never let facts get in the way of a good story. I was writing subjective nonfiction all along. Okay. But it is based on at least some experience and drawn from some of the experience of others as well. And it describes a research expedition that he ostensibly took, or someone took, or a collection of people took, into the Canadian north to investigate whether the wolves were trashing the caribou. Okay? And the way it's described in the book is it is as if the Canadian Wildlife Service had charged him with proving that they were doing it so that they could justify eradicating the wolves. The Canadian Wildlife Service pushed back strongly against that idea. But at any rate, he goes up and he finds out that the wolves, in fact, are living on mostly on little rodents. And when they do take after the caribou, what they're doing is culling the herd of the weaker animals. But he's a fabulous writer, okay? It's very amusing, very humorous. So I highly recommend the book, and I would read the book first, but then go see the movie. The movie stars Charles Martin Smith, who was Terry the Toad in American Graffiti and the sequel, and he does a fabulous job. It's all very well done.
Nice. Thank you.
Cool.
I was in that store, Kathy, once years ago. The
FAO Schwartz.
Yeah, it's very cool. You're muted.
Oh no, Kathy, you're muted.
Yes, I was too, and I muted because I'm trying to find that when the FAO Schwartz returned. So it shut in 2015, but it may have returned around 2018. I don't know.
Okay.
Alan, what do you have for us?
I have a little video that you can play on any web-connected device, especially nice here in your winter holidays. This is a NASA rocket engine fireplace video loop. So if you hit play on it, you'll get it's like a CGI image of a fireplace with a couple of little props around it and fireplace pokers and such next to it. And then in the fireplace, instead of a roaring fire of the Yule log, there is in fact an image, a video loop of the 4 RS-25 rocket engines and a pair of solid rocket boosters that launched the Artemis 1 rocket to the moon. And so that is just playing on a loop, and it makes a very nice fireplace. I'm sure it's very, very warm, too.
I feel like the Mingarelli household this Christmas would love that. I will definitely put that on.
Put it on an
TV.
iPad or a laptop, or if you have a big screen that's web
I do have a
connected,
TV.
you can just
TV's the
put
best.
this on. Yeah.
Okay, my pick is a webpage that tells the difference between a raven and a crow. Common raven is Corvus corax, and the American crow is Corvus brachyrhynchos. This morning I'm waiting for the train, and this beautiful big black bird is soaring around, right? He finds the highest point on a building, and it sits at the top looking down on the world. It's like, man, this is a bird that knows what it wants. And it's flying all over the place. There's a bridge that goes over the tracks. At one point, he goes on the bridge. He starts walking across the bridge like he owns the place. And he makes this weird noise. So I said, that's got to be a raven. It's not a crow because it's big and soars, right? So this page will tell you the difference between a raven and a crow.
I'm getting site not found.
Oh,
Yeah.
I got it. I found
Oh, I
it.
got it.
Oh,
Yeah,
it
too.
showed up for
It
you?
says
Yeah.
it says it's not secure though, the website, but
Yeah, I
but
got
I
a not
found it.
secure. Maybe it's because of my browser settings.
probably.
It's just a Washington nature program.
Yeah.
So it's
Okay.
about Washington, but it tells you the differences between the two. It has photos and talks about their feather differences. Like ravens have wedge-shaped tails and crows have fan-shaped tails, which is hard to see from the ground. And if you see a crow soaring for more than a few seconds, it's probably a raven. They can do a somersault in flight and even fly upside down. And they do gronk gronk, right? So crows go caw, and ravens go gronk gronk. And this is
Okay.
what this thing was doing this morning. It was sitting right next to the track, looking at everybody and making. It's a beautiful bird, really
The
big and
ravens are
dark.
so cool. They're so cool.
And the reason I look for this because there was an Instagram post this week about the difference between ravens and crows, which I thought was very good. And when I saw this, I said, I have to find. So this is a list of them. They say interestingly, you're not going to see them in urban areas. They're uncommon in populated urban areas, but I've always seen ravens.
I mean, you were in New Jersey, no? It's pretty populated.
Yeah.
It's very popular.
Did it say nevermore?
Yeah, nevermore. That's what I think of ravens all the time too. Raven is a great name, right?
Yes. There's
Like it.
also a small population of them in London, at the Tower of London. They live there.
I think that would be a great name for someone, Raven.
Sure. I'm
Right.
sure there are people named Raven.
I'm sure there are. It's just a really, really cool name, and the bird is very cool. They have personalities.
Laura and I discussed when we were about to become parents names that we could never give a child with my last name, and Raven was among that list.
Raven Dove.
That's true.
Yeah, you can't have Raven Dove. You can't do any
Yeah.
kind of wildlife name if you're a dove.
That's a total wildlife name.
Yeah.
Raven Dove. That's very funny. We have a listener pick from Jan. Dear Twivers, I was recently listening to some very old Twiv episodes, something down in the 200s or 300s. Several of you guys were saying that you played in marching bands back in the days. So I would suggest that you listen to the German techno marching band Mute. How do you say that, Kathy?
Not sure.
Okay, Mut, the Pack in German. These guys they take EDM songs, electronic dance music, and rework them and play them live as a full marching band. Literally everything these guys do is great, so it's difficult to suggest a track to start with. So Jan gives us a couple of YouTubes
Cool.
to look at. And if you have a little more time, I would suggest the rooftop concert on the Elbphilharmonie in Hamburg. And it's got a video for that too. A word of warning, this music is highly addictive, but it will put a big smile on your face.
Cute.
Keep up your great work.
It might be pronounced Moita
Moita.
like Hoita. What do you think, Rich?
I like that. I like that pronunciation.
Moita.
That's good. Yeah.
Okay.
Sounds right.
P.S. I studied in Marburg back in the 90s, and every time I hear about the virus, I get a pang of nostalgia. I was living just a kilometer away from the Bering work where the virus was first found. There was even a monthly culture magazine called Marburg Virus. Oh my gosh. Culture magazine. That's very cool. This marching band is just great. The EDM. Oh my gosh. I
I
love
haven't
what
listened
people
to it.
do.
I'm going to do that as soon as we close out.
I
here.
love what people do. That is TWiV 1179. You can find the show notes at microbe.tv/TWiV. You can send us questions. You can send us comments, pics. TWiV@microbe.tv. That's an email address. And we'd love to have your support, your financial support to continue these programs. Microbe.tv/contribute. Kathy Spindler is Professor Emerita at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Always a good time. Sure thing.
Somehow your affiliation got erased from the notes, but I memorized it
Well, you have said it a couple of times.
thousands of times. I can't believe we go way back.
I know.
So far back in time.
I know.
Oh, my gosh.
It's
Alan
very
Dove
cool.
is at alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
Angela Mingarelli is at McGill University. What's ImmuneVet? Where are you? Is that X or Blue Sky?
That's eggs. Yeah.
Thanks, Angela. Good to see you.
Yeah. Thank you and happy holidays.
Yeah.
Yeah.
Even though
You
this will
too.
come out after Christmas.
Yeah,
I
there's
know
a-
that all holidays, though. Any holiday you celebrate.
Yeah, this will be up before New Year's, so you can
Hmm.
have a happy New Year. I'm
Exactly.
Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.